The overall goal of this experiment is to assess the effects of a bacterial infection on the ability of host cells to form stress granules in response to exogenous stress.
This method is a valuable tool in the field of cellular microbiology for assessing whether or not a given pathogen is able to alter or subvert the host stress granule pathway. The main advantage of this technique is that stress granules can be qualitatively and quantitatively be analyzed in a rigorous and automated manner using free image analysis programs.
This method provides insight into the dynamics of stress granule formation and composition and how these parameters are affected by a specific pathogen.
Okay.
Before beginning the challenge, inoculate a red S. flexneri M90T bacteria colony from a freshly streaked tryptic soy agar 0.1% Congo red agar plate into a 15 milliliter conical tube containing 8 milliliters of tryptic soy broth. After tightening the lid, incubate the colony overnight with shaking at 222 rpm and 30 degrees Celsius. The next day, subculture 150 microliters of the bacteria in 8 milliliters of fresh tryptic soy broth for about 2 hours at 37 degrees Celsius and 222 rpm to initiate a late exponential phase induction of virulence gene expression and to enhance the bacterial infectability. When the optical density of the subculture is between 0.6 and 0.9, transfer 1 milliliter of the bacteria into a 1.5 milliliter tube for their collection by centrifugation, and resuspend the pellet in infection medium at an optical density of 1. Next, aspirate